Dear students, we have to continue the lesson, that is chapter 3, Chromosomal Basis of Inheritance, and which we have already started the chromosomal mutations or chromosomal aberrations, which includes change in the structure of chromosomes, change in the number of chromosomes. We started already change in number of chromosomes, what is called ploidy. Under the ploidy, we have discussed about aneuploid. Now the second type of ploidy, you see that one euploidy. So what is euploid? So an organism or somatic cell has one or more than two basic sets of chromosomes. That condition is called euploid. So it is a condition where the organisms possess one or more basic set of chromosomes. Either one set or more than two sets, like 3n, 4n, 5n, 6n, like that. That condition is called euploid. Entire set of chromosomes added for that condition. Now it is being divided into monoploidy, diploidy and polyploidy. You know very well about what is diploidy. So it is a condition in which an organism or a somatic cell has two basic sets of chromosomes. And that is called diploid condition. Now what is haploidy? Half the number of somatic chromosomes. Half the number of chromosomes of the somatic cells, we can say. Half the number of somatic chromosomes is referred to as what we have a gametic chromosome number, or we can say haploid. So the gametic chromosome number, you can find it only in the case of gametes, hence called gametic chromosome number. Diploid condition, in the case of gametes we have haploid condition. That is called haploid. We can get the haploid condition only in the case of gametes, not in the case of somatic cells, except in the case of certain plants. You know, we have the bryophytes. The gametophytes are n in nature. The cells contain n number of chromosomes. Okay, so we have the somatic chromosomes, half the number of somatic chromosomes, that is 2n, instead of 2n, that is n. And that is called gametic chromosome number because it is found only in the case of gametes like sperm or egg or the spores, etc. It's called haploid. But here we are talking about what is called monoploidy. Is there any difference between monoploidy and haploidy? Now, haploid condition is observed in the case of gametes. If the organism has haploid set of chromosomes in somatic cells, then it's called monoploidy. So, if the haploid set of chromosomes, n number of chromosomes is found in gametes, then it's called haploid condition. If n number of chromosomes is found in the somatic cells, that is called monoploidy. So anyway, you can see, now haploidy is different from monoploidy. Haploid condition is represented by n, that is the letter, and monoploidy is represented by x, the letter. Okay, so monoploidy condition is observed in somatic cells when you have n set of chromosomes. When you have the n set of chromosomes in the gametes, it's called haploidy. Now I'll give the example. I'll represent the example on the board now. To differentiate a monoploid condition from a haploid condition, I am taking the example of wheat. In common wheat, in the case of common wheat plant, which is a polyploid, that is hexaploidic condition. That means there are 6 sets of chromosomes. That 6 sets of chromosomes is equal to what is called a normal diploid. So here the diploid organism normally has 72 chromosomes. That means it has 6 sets of chromosomes. So 2n is equal to 72, but contains 6 sets of chromosomes. If you are taking the haploid organism, from that what we have this hexaploidy, n is equal to 36. N is equal to 36. But if you are taking the monoploid condition, that is x. So normally 6x is equal to 72, x is equal to 12. So this is not what is called haploid. This is a haploid condition because diploid condition we have 72, haploid condition we have 36, half of the chromosome. But if you are taking the monoploid with reference to the somatic cells, As we have 6 sets of chromosomes in the somatic cell, one set is equal to 12, and that is a monoploid condition. That is the difference between monoploid. So this 12 chromosome is referring to the number of chromosomes in a somatic cell. This haploid set, that is 36, referring to the number of chromosomes in a gamete. That's the difference. This is in somatic cells, and this is in the case of what we have actually the gametes. So this is what we can say, the difference between a monoploid and also what is called a haploid. Now, normally in a plant or an animal, the haploid and diploid conditions came regularly. And also, that's actually haploid and diploid conditions came regularly one after another. So diploid organism produces the gametes, that is M. Once again the diploid condition is restored during fertilization. That's why I'm writing like that. Haploid and diploid conditions came regularly one after another. So during the reproductive process. And the same chromosome number are maintained. See that one from generation to generation, either in the body cells or in the gametic cells. The same chromosome number are maintained just in the body cells as well as in the gametes, we can see. So the same chromosome number is maintained from generation to generation. Okay, so came regularly. But for monoploid condition, it occurs only when an organism is under polyploid condition, only under polyploidy. The meaning for that one just refers to the somatic cells. So actually, but monoploid occurs when an organism is under polyploidy condition. So polyploid, when you are talking about polyploidy, it refers to what is the somatic cells. More than 2 sets of chromosomes. That is called what is polyploidy. So the monoploid condition occurs or expressed only when an organism is under polyploid condition. We can say monoploid. That is called 1 set of chromosomes. Okay. So in a true diploid, if you are taking the diploid, both what we have the monoploid and haploid chromosome numbers are same. Either it is haploid or it is monoploid. They have only what is called 1 set of chromosomes. That is in a true diploid. Then a monoploid can be haploid. A monoploid can be haploid, having n set of chromosomes. But not all the haploids cannot be a monoploid. We cannot consider the haploid condition as a monoploid because it occurs only in the case of gametes. So we are talking about polyploidy and monoploidy with reference to only the somatic cells. If we are talking about monoploidy, what we have diploidy or what we have polyploid, all refer to what we have the somatic cells. But if we are talking about haploid condition, it's only the gametic cells. That is in the case of sperm or egg or in the case of spores. So anyway, a monoploid can be a haploid. That's a number of chromosomes. But all haploids cannot be monoploids. Okay, so monoploids referring to only the number of chromosomes in a body cell. That is X is equal to N. X is equal to N. So haploid set of chromosomes in a body cells is called monoploid. Haploid set of chromosomes in a gamete is called haploid. So monoploid is used only for the somatic cells. Haploid is used only for the gametic cells. That's why it's called as a gametic chromosome number. Okay, we can go for the different types of what we have, that is euploid. Now the flow chart shows the various types of euploid. So it is being divided into haploidy with n number of chromosomes. If it is present in the body cells or somatic cells, then it's called monoploidy. So haploidy in gametic cells, and monoploidy, that is called X in the case of somatic cells. Then the normal chromosome number, you know that 2 sets of what we have chromosome, that is called diploidy condition. Then if the organism has more than 2 sets of chromosomes, more than 2 sets of chromosomes in somatic cells and body cells, and that condition is called polyploidy, and the organisms are called polyploids. Now the polyploidy is further differentiated into autopolyploidy and then allopolyploidy. Auto means same. So organisms having the same sets of chromosomes, I represented A, all derived from within the same species. Then in the case of allopolyploidy, we have two different sets of chromosomes, AA and BB. It is being formed because of what is called interspecific cross. I will explain one by one later. Here just we have two dissimilar pair of chromosomes. Now this autopolyploid is further divided into autotriploid. This is based on the number of sets of chromosomes. Here there are 3 sets of chromosomes. There we have 4 sets of chromosomes. Accordingly it is called autotriploid. So triploid 3 sets, similar sets of chromosomes. 1A is equal to 1 set. And here also we have similar sets of chromosomes, all of what we have the similar sets. So they are called autotetraploid because of 4 sets of chromosomes in the body cells. I will explain one by one. This is the flow chart of the classification. Now let's go for what is polyploidy, what are the types of polyploidy and the different types according to the syllabus and the examples also, how can you get it artificially or naturally. Now what is polyploidy? It is a condition where an organism possesses more than 2 basic set of chromosomes. More than 2 basic set of chromosomes. For example, 2n plus n is going on according to the number. Now according to the number of basic sets, this is one type of classification. According to the number of basic sets, there are, so these are divided into the following types. These are divided into the following types. What are they? Number 1, triploid, having 3 sets of chromosomes. Tetraploid with 4 sets of chromosomes. Pentaploid with 5 sets of chromosomes. And hexaploid with 6 sets of chromosomes in the body cells, all referring to only the body cells. We are considering polyploidy only with reference to the chromosome number in the body cells. Then, normally if you are taking what is called polyploidy, it is very common in the case of plants and very rare in the case of animals. If it is observed in animals, it leads to the death of the animal. So, it causes lethality, death in the case of animals, but it is beneficial in the case of, for example, plants. For example, the tomato, the triploid tomato. So that is larger in size, that is different from the country tomato. Okay, but in animals, the polyploid condition always results in death. No animal with a polyploid condition, which leads to the death, even in the case of human beings also. So, normally an increased number of what is called sets. So it has been normally an important factor in the origin of new species. New plant species are formed because of increasing number of chromosome sets. But if it is going on increasing, it results in death. That's why written, but increase in ploidy level more than the limit leads to the death. So increase in number has been considered as an important factor in the origin of plant species. But if it is going on increasing the ploidy, then what will happen? Death results. Now what are the types of polyploidy? This is based on the origin. Now number 1, autopolyploidy. Number 2, allopolyploidy. So I will explain that one. So autopolyploidy, the condition is normally developed from the what is called similar sets of chromosomes from the same species. And this is a condition which is developed because of what is called the crossing between 2 different species. Okay, that's about autopolyploidy, allopolyploidy. We'll discuss more about these 2 things in detail. Now what is autopolyploidy? So it is a condition where an organism possesses more than 2 haploid set of chromosomes. These chromosomes are derived from within the same species. So they have derived the chromosomes from the same species. That condition is called autopolyploidy. See, I represented 1 letter is equal to 1 set. So this is about what we have. So organism which possesses more than 2 haploid set of chromosomes derived from within a same species, and such organisms are called autopolyploids. So if you are taking the types of autopolyploids, there are 2 types based on the number of sets. One is autotriploids, another one autotetraploids. I'll take the first one, autotriploids, and then I'll go for autotetraploids. See, autotriploids, I mentioned, you see that one, 3 sets. So these organisms have 3 sets of its own genomes. That is A only, not B. That's the meaning for own genomes, similar genomes. So these organisms have 3 sets of its own genomes. That condition is called autotriploids. Normally we can produce these autotriploids by means of artificial cross. They are not present in nature, they are being produced artificially by crossing, what is called actually by crossing between a tetraploid with the 4 sets of what we have autosomes, or we can say 4 sets of chromosomes in the somatic cells, and a diploid. Here I have given the illustration. Now this is the parent. What we have, this is the parent. So one is actually 2n condition, that is equal to 4x. Here 2n is equal to 4x. So, and another one, a diploid organism, 2n is equal to 2x. Now the gametes formed, this organism produces actually a gamete haploid number, but it is equal to 2x, 2 sets. And here it produces a haploid with only 1 set. So the crossing occurs between these 2 gametes results in the formation of a diploid organism, but the diploid condition is equal to 3 sets of autosomes, that is EE. But these individuals are sterile due to defective gamete formation because unequal number of sets. You see that one, it is fertile. This is actually also fertile because we have paired sets, so that gametic formation is normal. But if you have just actually 3 sets of autosomes, the gamete formation is not normal. So we need actually pairs. This is one pair, so that here 2 pairs, one and another one. So they need to have equal sets of chromosomes, then only gamete formation is possible, but here they have unequal number of sets. That's why they are sterile in nature. They cannot produce normal gametes. The sterility is due to defective gamete formation. The following are the examples of autotriploids. Number 1, cultivated banana. It is seedless, have larger fruits than diploids. The second one, sugar beet. So it has higher sugar content than diploids. This is number 1. Also this is resistant to molds. This is resistant to molds. That's a fungus. It is resistant to molds, a fungus. Then the next one, a common name, the bluegrass, Cyanodon dactylum. It is a natural autotriploid. It is not being induced artificially. Naturally occurring autotriploid is Cyanodon. Then others are, we have the tomato, the jam tomato, the apple, seedless apple, seedless watermelon is also an example for autotriploid. Sugar beet I already mentioned, and banana. These are all man-made autotriploids. That means they are being produced artificially. This is the one natural, and these are all the artificial ones, man-made, just actually autotriploids. Now the next one, autotetraploids. There we have 3 copies. Here these organisms have 4 copies of its own genome. 4 copies of its genome. How are they produced? They are produced by doubling the chromosome number of diploids. So if you are taking diploid organism, they are treated with what is called colchicine. So they are being, actually the chromosome number is being doubled. So it is 2n, and we have what we call the 4n. That's why they are called as tetraploids. So they are being induced artificially by chromosomal doubling with the help of colchicine, any mutagenic agent. Mostly we are using colchicine, a substance and alkaloid obtained from colchicum paternale, a plant. Okay, now, so what are the examples? So we have coffee, groundnut, seedless grapes, potato, alfalfa, another plant, and rye, the sickle cereal, a millet. These are all the examples for autotetraploids. So with that I concluded about the autoploids, autopolyploids. Then we have to go for just another one, what we have, that is allopolyploids. So what are allopolyploids? Some examples. What is allopolyploid? Organism which possesses more than two basic sets of chromosomes, but these chromosomes are derived from two different species. I represent species 1 and species 2. There you have derived from the same species. Now it is produced by means of interspecific processes between two different species, and the fertility is restored by doubling the chromosome number. See here, if you are taking for example, this is a F1 hybrid. By means of what is called colchicine treatment, now the chromosome being doubled. So we are getting pair for each one. So the fertility is restored normally by chromosome doubling by treatment with what is called a mutagenic agent colchicine. Now they are produced only between closely related species. Examples are Raphanobrossica, a cross between what we have radish and then what we have just is cabbage. This is a cereal obtained from what is called wheat and rye. I mentioned two examples for allopolyploids. One is Raphanobrossica. So a Russian geneticist by name G.D. Karpentenko in 1927 crossed what is called radish. The binomial for that one, Raphanus sativus, having diploid number 18, with cabbage, Brassica odoraceae, that also has what we have 18 chromosomes in the body cells, and produce F1 hybrid, but the F1 hybrid was normally sterile. This is because of defective gamete formation. When he just doubled the chromosome number by treating the plant with colchicine, he got it fertile. So normally what we have, he expected the plant to exhibit the character of radish, that is a root of radish, and the leaves of cabbage. But the result obtained was vice versa. Okay, that's why he was normally got disappointed. Now here is a cross I given. So Raphanus, what we have the radish with the 18 chromosome is crossed with the Brassica, that is a cabbage. It also has 18. So parents. Now these are what we have, the plant fruits. Now the gametes form in each case with the N 9 chromosome. There are also 9 chromosomes. Now after fertilization, he received F1 hybrid, but the hybrid was sterile, having what we have 18 chromosomes after the union of gametes. Now this is sterile because of defective gamete formation. It is unable to produce normal functional gamete. When he treated the plant with colchicine, so he got the chromosomal doubling, resulting in the formation of Raphanobrossica, which is nothing but amphidiploid. Amphidiploid is another name of what is called allopolyploid. Amphidiploid is another name of allopolyploid. So it is having diploid number of chromosomes that's equal to 2n is equal to 4x, 4 is equal to 36, because each x, that is a monoploid condition, equal to 9, 4 into 9, 36. But the organism is considered a diploid, but having 4 sets of chromosomes, different chromosomal sets. Actually, see for example, like this. This is the nature of what is called the genotype. So two unequal actually genomes are present, but pairing is possible because we have two, there is A and two B's. So pairing is possible, so they produce what is called actually the fertile gametes. This individual is fertile in nature. Okay, amphidiploid is another name for allopolyploid. Now the second example for allopolyploids is cereal, what we have actually triticale. So it is a successful first man-made cereal, Based on the ploidy level, the triticale, the cereal, allopolyploid is divided into 3 main groups because of the combinations. 1, tetraploid. It is a cross between diploid wheat and rye. When we are crossing a diploid wheat and rye, we are getting what is called tetraploid. The second one, hexaploid, when you are crossing a tetraploid wheat, what is called Triticum durum. Triticum durum, normal name, the common name, macaroni wheat. And right, then results in hexaploid, cross between tetraploid wheat, Triticum durum, and right, that results in hexaploid. Then octaploid, third one, a cross between hexaploid wheat, what is called Triticum aestivum, which is commonly called as a bread wheat, and right, that results in octaploid. Now, I take one example. So the tritical hexaploid, this one, the tritical hexaploid hybrid. When I am taking the tritical hexaploid hybrid, that one demonstrates the characteristics of both macaroni wheat as well as rye. For example, they combine what these two actually, the plants combine. That is higher protein content of wheat with the rice, high content of amino acid lysine, because the lysine is very low in the case of, for example, this wheat. So the combination of both the characters, high protein content of wheat and also we have high content of lysine amino acid from rye, both are combined together in the case of what is called triticale. So I represent the cross. How is it produced? I am taking what is called the hexaploid, a cross between a tetraploid, namely Triticum durum, and the rye, what is called sickle cereal. And I'll represent the illustration. The hexaploid triticale hybrid is produced by making a cross between, that is what is called a macaroni wheat, Triticum durum, and a rye, what is called sickle cereal. Now in the case of macaroni wheat, the diploid number 2n is equal to 4x, that is 28. In the case of rye, 2n is equal to 2x is equal to 14. So this is a tetraploid and that is a diploid. Now the gametes formation, n is equal to 2x, that is 14. And here n is equal to x, that is 7. So the union of these two gametes result in a triploid. The triploid is sterile nature. This triploid is sterile. Suppose I mention here F1 hybrid, that is sterile. This is F1 hybrid, that is sterile, which is a triploid because of defective gamete formation. When it is treated with what we call colchicine, it resulted in chromosome doubling. So the 2n, just we have 4n, it is being doubled. 3x is being doubled to 6x. So 6 into 7, 42. This is a triticale, a hybrid. That is the first man-made cereal, which is a hexaploid. Okay, that's about the illustration showing how far the Triticum durum and sickle cereal combine together to produce a hybrid triticale. What is the significance of ploid? The polyploids are more vigorous and more adaptable to any environment. More vigorous means they produce large sized fruit, leaves, that is vegetables, etc. Now we have the ornamental tetraploid plants. Many are ornamental tetraploid plants. They have larger flowers. They produce larger flowers and also large flowering duration. The longer flowering duration. So ornamental autotetraploid plants have larger flowers and longer flowering duration. Now the autopolyploids, they have higher fresh weight. The fresh weight is due to the presence of more water content. So they have higher fresh weight. This is due to the presence of more water content. Now we have the aneuploids, where there is a loss or gain of different chromosomes. So through aneuploids, actually you can determine the phenotypic effects due to loss or gain of different chromosomes. So because of loss or gain, we have different phenotypic effects seen externally, we can find out. Then many angiosperms are allopolyploids, and these allopolyploids play an important role in evolution. So the role of evolution, phenotypic effects can be found, higher fresh weight, and also there is what we have the ornamental plants, the role of ploidy in that plants, and then we have more vigorous growth and more adaptable characters than what we have the diploid plants. So with this I concluded about what we have change in number of chromosomes. That's what is called ploidy. What are the types of ploidy, etc. We have to go for the chromosomal structural changes. We'll do it in the next class. Okay, thank you.